It's recording. Okay, so we discussed before the fact that the receptors in the eye and the signaling in the eye is kind of unique because unlike other receptors where the stimulus will cause depolarization and the neurotransmitter is released, this is not the case here in the eye, right? We agreed that the default in the darkness that the cells, the receptor is depolarized, right? And the default is that we have a current of sodium keeps coming in, right? And that causes the cell to be in constant depolarization during darkness. And that causes the release of the glutamate, right? So we have two scenarios here. So number one is during dark, right? And if we think of the receptor, the membrane for either the rods or the cones, that it has over here, there will be a channel, a very specific channel for sodium, and that channel is kept open by binding to something called cyclic GMP, right? It's kept open by binding to cyclic GMP. And that allows the sodium to keep coming in, causing depolarization of the cell, right? When the cell is depolarized, at the other end, where the synapse is, right? Where it synapses with the bipolar cells, then it will release from the other end. Let's just simplify and let's imagine that this is the synaptic end, right? And so from this end here, it's gonna start to secrete or exocytose a neurotransmitter called glutamate, right? We agreed to this, right? So this happens during darkness, right? So let's evaluate what happens when this receptor is subjected to light, okay? When it is subjected to light, we have here the rhodopsin, and the rhodopsin has the opsin protein, and inside it we have a dye called the retinal, right? Which is a derivative of vitamin A, retinol, right? So when you subject that to a light signal coming to it, that cis retinal, the cis retinal, is going to change the shape, right? Because of exposure to light. And that will make this structure unstable. And it will change the cis retinal to be something called all trans retinal. Sounds good? Because of this change in the structure, now this has become active and it starts slapping things around because it changed the structure of the protein. The rhodopsin now has changed, right? Okay. And with the change, then it's going to stimulate a protein residing right next to it. And if you remember, we call that protein transducin, right? As you slap it around, sort of like the G protein, remember the G protein that we have studied at some point, it also has an alpha subunit, and the alpha subunit is going to travel. But this time, this alpha subunit is not going to activate adenylate cyclase. Remember, in the normal signaling, the alpha subunit of the G protein activated the adenylate cyclase or the adenylate cyclase, and that resulted in cyclic AMP accumulation. This is not the case here. Matter of fact, it's going to activate a complex protein here called PDE, phosphodiesterase. And that phosphodiesterase is capable of breaking cyclic GMP into just GMP, right? So now you broke the cyclic GMP, right? Making sense? When you break the cyclic GMP, what happens to this channel? What do you think? You have less cyclic GMP. So what do you think would happen here? It's going to close, right? And therefore, the sodium current, or what we call the dark current, is not going to exist anymore, right? And so the cell is going to be less depolarized. And if the cell is less depolarized, so what do you think will happen to the glutamate? It will go down, right? So unlike other receptors, where the stimulation of the receptor will result in the release of the neurotransmitter, the photoreceptors, the stimulation will result in cessation of the glutamate secretion. Does it make sense? You stop secreting the glutamate. Yes? Okay. So that's the first part we dealt with, which is the receptor signaling, right? The action potential or whatnot inside the receptor, or what happens, the depolarization and all that. Yes?
What did you say about the
Phosphodiesterase. PDA is phosphodiesterase, which also breaks cyclic AMP as well, but different subunit of it will break it. We're going to study again the PDE, the phosphodiesterase, when we get to the reproductive physiology, because one of the best seller drug out there is actually an inhibitor to PDE. You guess what it is? I already told you it's reproduction physiology. Viagra. And so it's Cialis, which is kind of the same, and it acts through inhibition of PDE. So we're going to revisit that later on. Sounds good? Okay. So we're good with this? Yes? And you have that slide on your presentation. Now the on-off. You don't have to, for the exam purpose, you don't have to remember every single detail that we are describing here. But in case if you're interested, then this is how the on-off signaling works. And we need to understand the basics of the on-off. Why do we have the on-off? First of all, why do we have the on-off? Imagine your eyes as tiny LED spots, right? And if they light, right? If they light, the next door area will get some of the light as well, right? But that will blur your vision, right? You would like to say, okay, only this is the area that is lit, and everything around that area is blackened, right? So you try to isolate the light to know for sure that the light ends here. Making sense? Yes? Okay. So this is how our brain functions in order for us to understand, because this is not something like your touch, where you can say, I think it's coming from this particular spot in my finger. This is different, right? And the vision, you have to know precisely which receptor is firing, because this will tell you how to read particularly small text. Because you want to know that this point is different than that point. If the 2 points are bleeding to each other, that will not work very well, right? So what do? You isolate it, and you isolate it. They think this isolation thing works this way. So you have an area that corresponds in your brain, right? And that area will have a center, so you have something that looks like this, right? And so this will be what they think is the on center, and this will be the off center, right? Which means when this is lit, this shuts down, right? Therefore, in the brain, in the coding of the signal, will create an isolation of this point so it doesn't bleed with another point, right? So this one, if they are too close to each other, it's just on, and if this is lit, you may misunderstand that this is also lit, right? So you try to isolate it with a zone that is off, that tells you, no, nothing is happening. Make sense? Okay. So in this case, if the light is on, so now we have light, and this we have dark, right? When there is light, you would like this one to fire, and this one not to fire. Does it make sense? Right? So you're firing the center, and you're not firing the periphery of that area, not in your eye, but in the brain. These are 2 neurons in your brain. Making sense? Okay? And when it's dark, you would like this area to fire, but not this area, right? And we compared it last time to having a stakeout, right? Where two police officers or two detectives are standing at the street corner. One of them, his job is to tell you if the suspect is coming, and the other one to tell you that everything is good, it's quiet in here, right? And so you're getting both signals, right? So the job of this zone is to tell you, hey, it's dark. We're not seeing anything, it's dark, okay? And this one, in this case, tells you there is no light. You see, there is no light, that's an absence of a signal. But it's dark, that's a presence of a signal, right? So with that, that means every receptor in your eye has to fire two different kind of signals, right? One of them to tell your brain that there is no signal if there is dark, right? That there is no signal, and the other one tells your brain it's dark. You see, there is no light, and the other one says it's dark. Making sense? And then the opposite, if you have the light, then one will tell your brain that there is light, and the other one will tell your brain it's not dark anymore. You understand? Yes? Okay. So how does that happen? How does this signaling take place? We agreed that if we have a receptor, and here is a rod, and the rod, we agreed, it secretes to us neurotransmitter. We call that glutamate, right? You remember that? Does it secrete the glutamate during the dark or during light? You remember? During dark, and we call that the dark current, right? And so the glutamate now is being secreted, and so we will describe this as in dark. Why is it in dark? Because we have the glutamate. If it was in the light, then there will be no glutamate, right? Yes? Okay. And so we're going to have two kinds of, the next cells are called bipolar cells, right? So we have a bipolar cell here and a bipolar cell there. And those will feed to the next cell. Do you remember what's the next cell here? We call it ganglion cells, right? The ganglion cell. And the ganglion cell are actually forming your optic nerve, right? Yes? Okay. Occasionally, we will have also some horizontal cells that bridge those together, and they will help you in the lateral inhibition that we kind of talked about in the afferent signaling, right? And the precise look. Remember the story of the bird that tries to eliminate the competition? The horizontal cells are capable of doing that. Yes? Okay. So far we're good. We agreed to the fact that the glutamate is being secreted. Why is it being secreted? Because it's dark. And instead of having just one bipolar, you have two bipolar cells, right? It happens that one of the bipolar cells will have a glutamate receptor that stimulates the cell, right? So it causes depolarization, right? So they will differ from each other in the type of the glutamate receptor, right? And so in this case here, we will have a glutamate, and I'm going to call it just G, this is my own making up a name, GS, which is glutamate or GRS. This is, I'm making it up, right? So glutamate receptor, and it's stimulatory, glutamate receptor, right? On the other one, I will have GRI. Making sense? So I have a glutamate receptor here that is stimulatory, and a glutamate receptor here that is inhibitory. Making sense? Yes? Well, let's look at the glutamate receptor that is stimulatory, right? What will it cause? Depolarization, right? It causes excitatory postsynaptic potential, right? The bipolars are neurons, right? But these are one of the few neurons in your body that doesn't have action potential. Because it's so short, you don't need action potential. The whole thing can be transmitted with graded potential. Who needs action potential when you can just cross no problem, because it's just like few microns, right? It's not the feet that you have with the regular neurons. Sounds good? Okay. So let's talk about the stimulatory one. Glutamate will bind to the receptor, causing depolarization, right? And so now this will be activated. This will be activated. It's gonna send a depolarization wave to this ganglionic cell, and that will go to your optic cortex, to your visual cortex in the occipital lobe, to tell it, listen, we are receiving a signal that it's dark. Making sense? So you're actually actively sending a signal to tell the brain it's dark. So that's the off, right? You're activating the off in this case because it's dark. In the dark, you're activating the off. You're depolarizing the off. Are we good? Okay. So let's look at the other end. We have an inhibitory one. The inhibitory one will let potassium N, which we all know will cause hyperpolarization, right, of this bipolar. When this is hyperpolarized, do you think it's going to send any signal? Is it going to send any depolarization wave to this ganglion? Is it going to secrete anything when you're hyperpolarized? No. It's going to stop sending any signal to this ganglion. And this ganglion is the one that tells you there is a light, right? It's the on, right? So this one is the on. This one is the off. Again, the off will tell you it's dark. The on will tell you it's light. Making sense? Good. So now that you don't stimulate this, does your brain understand that there is a light? No. When you don't stimulate, remember this is inhibitory, so now we have inhibition, right? When you don't stimulate this one, does your brain translate that as there is a light? No. It will say the light is absent. Why is it absent? Because I don't have depolarization here, right? But when you depolarize this, what does this tell the brain? It's written here. It's off. It tells the brain that it's dark. So you're receiving two signals. One, from the same receptor, right? One that tells the brain that spot, not the whole brain, but that just spot, that it's off, and the other one that tells you that it's on, right? So you would think that means I'm switching on and off the light, right? This is what on and off you would think of. No. If you have a dark dot and next to it is a white dot, the dark dot is falling on a receptor that will translate that as dark, right? Because you're not receiving any stimulus from that black spot. The next to it, the area right next to it, which is white, is sending an active signal because now there is a light being emitted. Black is the absence of a stimulus. Black itself is the absence of light coming to you, right? The spot right next to it is actually firing, right? So now you have one with on-off and the other one is on and off. How do you think we are able to read text? This is it, right? So there is one spot that sees a dot or a letter, right? And the other one right next to it can't see anything, right? Because one is seeing the light, so you're not in vision. Let me rephrase that. You're not seeing the letters. You would think you're seeing the letters. You're seeing the absence of a signal that's coming from the black area, right? You're seeing the absence of the letters. You're not seeing the letters. Does it make sense? If the letters are black on a white background, you're not seeing the letters, but you're seeing the absence of a signal from the white board. Make sense? Okay. Anyways, back here. We understand what is the off and the on. Let's have another scenario and you will answer me in this case here. Let's say that this now is light. And the light, again, doesn't have to be universal light where you turned on the light. No, this receptor is receiving a signal, right? So now you're moving your eye from a black spot to a white spot. Now the receptor is receiving a signal. Or it's global, where you turn on the light or turn off the light. Either way. Make sense? Yes? Okay. So now we have light, right? When you activate the receptor, what happens? We agreed the phosphodiesterase will be up, transducin will be activated, phosphodiesterase will be activated, therefore cyclic GMP will be down, and therefore the sodium will stop coming in, right? Causing the depolarization. So now you don't have glutamate. So now we are lacking the glutamate. Are we good? Okay. If we lack the glutamate, remember the glutamate in this case was causing hyperpolarization. Yes? Hyperpolarization, because it's inhibitory, right? And now I don't have the hyperpolarization anymore. So this one will start firing, because I removed the hyperpolarization. And so therefore this ganglion cell is going to be activated to tell your brain, hey, there is light. Yes? Let's look at the other end. This, when we stimulated it with the glutamate, it told your brain that it was dark. But now you're not stimulating it anymore with the glutamate, so it's going to fail to tell your brain that it's dark. Get the point? So you'll have one ganglion cell that will tell your brain that it's light, in this case, and the other one will fail to tell your brain that it's dark. Make sense? And now you have the on-off switch that we talked about. Make sense? Yes? Okay. Now for the exam, we don't necessarily need to really have this elaborate conversation, but this explains hopefully to you. And this will be posted to you so you can repeat it, but don't get too obsessed about it. This is going to be maybe 1 or 2 questions in 100 question exam. So just, you understand, right? We're good? I'm going to turn this off now.